Hello everyone, my name is Kong Shurnani and welcome to my YouTube channel NeuroChat with AG. On this channel, I plan to cover topics that are closest to my heart and support the neurodiverse community. Topics related to neurodiversity, autism, late autism diagnosis, and autism in women and girls. I will interview thought leaders, esteemed researchers, scientists, professors, doctors, and psychologists and seek their views and updates on their latest work and research. I will also chat with parents of neurodiverse kids and adults and talk about experiences of neurodiverse individuals. Lastly, keeping inclusivity as a topmost priority, we might also discuss controversial disparate views for a collective understanding of issues and respect for diverse views. All the views and topics discussed are for informational purposes only. If you are interested in watching more quality content on this topic, please hit the subscribe button. Don't forget to press the bell icon to be notified whenever a new video is uploaded. Please also share the channel with your friends, family, and social media platforms. Thank you. Dr. Juergen Hahn is a distinguished researcher and academic leader. He currently serves as a professor and department head of biomedical engineering at Rensselaer Polytechnic Institute. He is renowned for pioneering work in systems biology, computational biology, and data-driven methods in healthcare. One of his most notable achievements is the development of non-invasive diagnostic tools for autism using advanced data and analytics. By analyzing metabolic and biochemical profiles, Dr. Hahn's work has identified key biomarkers that could enable earlier and more accurate autism diagnosis. This research has the potential to revolutionize the diagnostic process, reducing reliance on behavioral assessments and paving the way for personalized intervention strategies. His studies have also exposed the role of the folate-dependent 1-carbon metabolism and transsulfuration pathways in autism, providing insights into the biochemical underpinnings of the condition. This has contributed to a deeper understanding of autism with biological, neurological, and environmental influences. Dr. Hahn has published extensively in high-impact journals and is recognized for bridging engineering, biology, and medicine to develop innovative solutions for early diagnosis and personalized treatment strategies. Dr. Hahn, welcome, and thank you very much for the time today.
Well, thank you for having me.
And to jump right into it, can you please share a bit about your background and your professional journey?
Oh, of course. So I got originally my undergraduate degree from Aachen in Germany, and that was back in 1997. I then went to the US for graduate school. I was at the University of Texas at Austin, where I received my PhD. And so by training, so my degree is actually in chemical engineering, but I'm a systems engineer by training. Okay, so I always looked at systems concepts. I did a postdoctoral stint in Germany and joined Texas A&M University as an assistant professor back in 2003. And then I moved to Rensselaer Polytechnic Institute. I will usually call it RPI, just so people know the acronym, in 2012. And I joined the Biomedical Engineering department, given that all of my work really focused not just on systems, but really on biological systems. So systems biology is kind of the key word, how I would describe most of my work. And I became department head in 2013. And I'm currently still a department head, but on my way out, given that I took on a different role at the university, but I'm obviously still research active and run an active group. So that's kind of how I got here.
Your research, which integrates systems biology with computational models to study diseases and disorders. So regarding that research, could you explain how computational biology can provide insights into complex diseases that traditional methods might miss?
Okay, let me start back where I'm originally coming from, and then I kind of answer your question in the long way. Okay. Like for anybody who does drug development, there's certain targets that you have, and there are oftentimes some metabolite that you're trying to monitor, and you want to affect this metabolite with your drug. And what you find is that you can find some chemical compound that, let's say, lowers or raises the concentration of this target that you have, and that works really well if you just look at this one-on-one, but given all the complexity that you have in human cells, is that when I only affect this one compound, your body tries to compensate somehow because it's part of a biological network. Okay, and so just looking And what you find is that most of the compounds that you identify that do this actually don't do the job properly, because your body says, well, this is lower than we want it to be, even though that's exactly what from a pharma development you want to do. And instead says, I need to compensate for this, and I'm going to bring up this level of this one compound again. Yeah. And what we do in systems biology, we look at the networks as a whole. So it's a more holistic approach. And we basically take into account, like, just knocking out like one reaction that leads to the formation of this compound may not be enough if the body tries to compensate for it. So we're looking for more overall patterns on what could be done there in order to basically get the outcome that you want. Now, getting into how can you use this for disease diagnostics, okay? Let's pick an example. I don't want to make this sound like this is a simple example, but if you diagnose diabetes, okay, the diagnosis of diabetes is done by measuring somebody's blood sugar. Okay, now you can do this by the fasting blood sugar in the morning, or you can take the A1C, which is a more average value for blood sugar over a 3 months period. But you look at this, if it's too high, you would diagnose somebody as having diabetes or pre-diabetes or not having either of those. Yeah, so use one measurement to determine this. Yeah, let's get something that is a bit harder to diagnose. Let's say heart disease. There we're measuring cholesterol, but you're using more than one measurement to determine if somebody is at risk of heart disease. Like you look at, you have good cholesterol, you have bad cholesterol, yeah, so the HDL and the LDL. But we're also looking at the ratio, for example, yeah, and you're looking at the total, which is the sum of two. So now we're using two measurements and some mathematical combinations of them, and you take all of this into account to determine if somebody has a high risk for developing heart disease or not. Yeah, but it's already different from what I just used for the diabetes. We're just using one measurement, yeah, or one measurement over time. So now if you're looking at much more complex conditions, and I'm going to throw autism into that mix, that you're going to need a lot more than two measurements. Okay, you need to basically, autism affects so many different physiological systems in the body that you're not going to have a diagnosis based upon one measurement or even two. You're going to have to look really at multiple ones. That means you're going to have to look at patterns of these measurements that you have, yeah, because it could be that they're the sum of two or the ratio of two, or it may even be a combination of 5 or 6 of them in some form. And that's really where the computational aspects and the data science aspects come in, because it's all about, can I determine this is a pattern, let's say a pattern of metabolite measurements that come from somebody who has an autism diagnosis, and in comparison, we have the same measurements from somebody who doesn't. Do I find differences in patterns there when I look at the picture as a whole? So if I take, for example, 2 blood samples where somebody one comes from somebody with an autism diagnosis, another one from somebody without, you're not going to find a single measurement that tells you, oh, this sample came from this person and this sample came from the other. You're not going to be able to do that. But you may be able to find these patterns where saying, hey, this looks very different in people who have an autism diagnosis compared to others that don't. And this is kind of where you marry the data science approaches with the human physiology in order to use this for diagnostic purposes. So very long answer, but hopefully eventually I got where you wanted me to be.
Yeah, that was great. And your work intersects with neurodevelopmental disorders like autism. And so what are some of the key findings you have from your studies on autism?
Okay, so as I said, coming back to where I originally came from. Yeah, as I said, I've always looked at computational systems biology. We've did a lot of work 20 years or 15 years, 20 years ago on inflammatory bowel disease and inflammation, but none of this had to do with developmental disorders. Yeah, but it's the same type of algorithm that you're developing and using there. What really got me excited about applying these things to autism was when I read some of the earlier studies where people looked at metabolites, in particular the folate 1-carbon metabolism. Yeah, and we noticed that, look, they're taking the same measurements. People are looking at the pathway as a whole, but the analysis was still done on let's compare their statistical significance in individual levels. Yeah, and so we said, well, we have all these algorithms laying around. Why don't we apply them here and see what you can get from this? In comparison, when you look at the state of the art for diagnosis, the state of the art is all by observations. Yeah, I mean, people basically study or people fill out questionnaires and observe how somebody behaves, how they interact with people, how they interact with toys, and then based on these observations make conclusions. But given my background, I like to have some biology and physiology in our work. Yeah, and so we said, like, look, let's see if we can also find differences than when we're looking at the biological pathways. And this kind of what got us excited about it. And what was interesting, some of our earlier work showed that there are very significant differences in some of the metabolites, especially if you're looking at metabolite patterns. And when we published some of this work, the feedback we got was literally, honestly, it was overwhelming compared to the feedback I got before. I mean, usually you had you had relatively few people who were super excited about your work. Yeah, you got somebody who cites your work, but they're all in the scientific community. But suddenly we got a lot of feedback from people saying, hey, this is really something that's very different from what's currently being done. And we really would like to see more of this. And this is kind of how we got more into more working in the autism space. And for the last decade, my entire group is really just has applications in related to autism. So this is how we got there. I would argue that pretty much any of these conditions that are diagnosed by observations, and that's not just autism right now, that you're going to find some biological underpinnings of these if you basically just look deep enough and you use the state-of-the-art tools for doing that. Yeah, my focus is on autism. That's where my collaborators are, and that's where our background is. But I would not be surprised if very similar approaches can be used to a variety of other conditions. It just hasn't been done yet for the most part. But that's kind of how I got into this field, and I do think there's a lot of relevance. But of course, it takes a mindset change for people to accept that there is a biological underpinnings to a lot of this, and it is not that ultimately the changes in behavior that you're seeing is really more a result of the biology rather than that it's independent.
Thank you. And your long-term goal is to improve the diagnostic accuracy of autism by providing a biological basis for it. So how does this like, I guess, really differ from traditional methods? What are kind of the advantages of it?
So, I mean, there's multiple, but please keep in mind that a lot of the work we're doing is still very fundamental and hasn't made it into practice. Yeah, but so let's start with, if you talk about, so when do you want to diagnose autism? And most people would argue that at the 18 to 24 months age, that's usually the best time you diagnose it. It's not clear if you could diagnose it earlier. Yeah, and but at the same time, if you diagnose it later, in like things like early intervention services that you could have started, you kind of lose out on that if the diagnosis happens later. Yeah, but currently the average age for somebody to get an autism diagnosis in the US is over 4 years. Okay, so 4 years is quite a bit away from this 18 to 24 months window that we want to get to. If you look at other countries, they aren't doing any better. So it isn't just, you can say, okay, it's the US healthcare system where there isn't much screening. If I go to the UK, the average age is over 5 years. Yeah, and so in many, many countries, the average age is even higher than what we have here. So if you talking about earlier diagnosis, then you find that there's many, many different factors that play a role in this. Okay, there's ranges from that not everybody necessarily has access to the healthcare they need. Yeah, and especially if you live in either poor areas or in very rural areas. But the other problem is also diagnosing 18 months old is hard. Okay, you may find that, well, I mean, some people may be delayed, but they may not have autism at all. Okay, the kids may develop very differently, so it's hard to diagnose many kids that young. And so often, what I've heard over and over again from parents is like saying, "Well, we had an earlier visit to a specialist, and they had concerns, but they said, well, let's wait another 6-12 months, come back, and let's look at this again." And as things did not improve, then they got the diagnosis later. But the key question is, so could you give people more data earlier that would allow them to saying, okay, we have a higher confidence that this person does have an autism diagnosis, and maybe they should start some early intervention, yeah, to basically figure out what the needs are. Yeah, and so anything that gives another data point to somebody who performs this evaluation can be very, very useful. And that's kind of what we're getting into. Now, there's always this argument, okay, outside of the 18 to 24 months window, could we diagnose this at 12 months? Could you diagnose it at 6 months? Could you diagnose it at birth, right? When we're looking at just biology, like blood samples is most of the work that we do, is that sure, I can take a blood sample at birth, but I don't know if I would find what I need to find there. Unfortunately, these studies are really, really hard to conduct, because the difference is, if they're old enough, I usually know if somebody has a diagnosis, and so I can recruit somebody as either having a diagnosis or not. If people are below the age where they could even have a diagnosis, then you basically would have to recruit a much larger number of people, and you have to follow them later on and ask, well, did you get the diagnosis later on? So these studies are very hard to do. I don't know if our method would work if you go to much younger kids. Okay, it may or may not work. Yeah, but nobody's done that, and but it's possible that it could work. Let's say that, but. It's work that nobody has done, and therefore we don't have an answer there yet. But regardless of that, if you can get your average diagnostic age down from the 4 years that it's now to 2 years or a little below that, that would be a huge win for everyone. And that's kind of what we're looking at. And because that one, we know from our work, it certainly seems to work in the lab.
Could you also please elaborate on the role of advanced data analytics in identifying biomarkers for autism?
Sure. I mean, look, I can tell you is that the samples we're looking at, if you look at individual metabolites, yeah, you're not going to find that basically that you can draw any conclusions from that. If I look at, let's say, as one example, if I just look at homocysteine levels, somebody with an autism diagnosis, somebody who doesn't, this doesn't get you anywhere. Okay, you have to look at that's what's called univariate statistics. Yeah, we're looking at one at a time. That unfortunately it doesn't work. Okay, and the main reason it doesn't work is that people who basically don't have autism, you have a huge heterogeneity on what is a normal level, okay, of these compounds. Okay, so this is that you may find that on average one group has a higher level, but that doesn't tell you anything about the individual. But the diagnostics is about each individual at a time. Yeah. But when you start looking at data science, we're going to ignore that there's 1 or 2 or 3 individual measurements, but we're looking for patterns of these. And suddenly these patterns look very, very different. So you have to use data science tools to look at that. Note of caution here, yeah. A lot of the data science we have is very much driven by software and algorithms that people have developed, mainly for things like web search engines and self-driving cars. Now we start using some of these same techniques to diagnose conditions. It's not the same, okay? So actually these algorithms have to be adapted, and this is where our work comes in to make sure is that how do we tailor this? So just using it out of the box isn't always optimal, okay? But the long story short is, you are looking for patterns and differences in metabolite concentrations rather than looking at individual ones, because that doesn't get you anywhere. The condition is simply too complex, and the presentation is way too heterogeneous to find anything by looking at things one at a time.
I was also reading about the profiling of blood samples of individuals with autism. So these signature researches could potentially serve as biomarkers for diagnosing autism. So, yeah, a little bit about that, please.
Of course. So, I mean, as I said, what we found in some of our studies is that basically, when I look at a certain number of metabolites, yeah, and these come from a blood sample taken from people with autism, and then I also have blood samples from people who do not have autism diagnosis. What we found is that we can predict with over 95% accuracy if this comes, the sample comes from somebody who has an autism diagnosis versus somebody who doesn't. Okay. Usually for diagnostic accuracy, people looking at 80% plus. Okay, so anything above 80% is considered good. In fact, the ADOS that people use, usually ADOS for 18 to 24 months old, you're looking at like around 80% accuracy. Yeah, and so basically analyzing these blood samples and telling you with 95% plus probability where they came from, that is certainly above the threshold what you need there. Keep in mind a couple items there is that number one, yes, we've done several studies on this, and I can tell you it certainly works in the lab. Okay, so there's in order to get this approved, you basically have to meet some other standards as well, and that requires more work that has not been done yet. So that's the one thing. The second item I want to say there, and this is a problem that we're currently working on, is that oftentimes people look at, can I distinguish somebody with an autism diagnosis from somebody who doesn't have one? And that's fine, but clinically, that's really not the point. Okay, if you evaluate, you don't randomly evaluate people for autism diagnosis. Yeah, people are evaluated means there are certain concerns in their development. Yeah, otherwise you wouldn't evaluate them in the first place. And so the question then doesn't become, is it autism versus not autism, but it's really comes, is it autism versus some developmental delay that is not autism? Okay, which is a much different question, because if somebody comes for an evaluation, you know their concerns, so they have some developmental delays. You just don't know, is it autism or is it something else? Yeah, and that is a completely different study actually, because now your control group is not typically developing people, but it's developmentally delayed children, but who end up not with the autism diagnosis. And so that's a very different study, and that's something that also needs to be done for this to be clinically relevant. And it is a point that we're looking at into.
Another thing that was very interesting that wasn't in my questions, but this is something that you talked about right before we started recording. So kind of like, I guess the importance of getting a diagnosis and how it can really help people, not just because of autism, but because of all the co-occurring conditions. So, if you could talk a little bit more about that on the recording.
Oh, absolutely. I mean, so look, the thing is, the autism diagnosis usually doesn't happen in a vacuum. Okay, there's plenty of statistics that show that people who are diagnosed with autism have a number of co-occurring conditions. They're much more likely to have certain conditions than they would have if they wouldn't have an autism diagnosis. Like one example, if you have an autism diagnosis, your risk of having epilepsy is tenfold, much, much higher. Okay, there's a very high percentage of people with the autism diagnosis who have GI problems, and these are GI problems that you can't necessarily fix using traditional means. I mean, this kind of spun off some work that other people are doing, looking at like the microbiome in autism, for example. Yeah, and but these are just two examples. Like, I mean, people with autism are much more likely to have sleep disorders. Yeah, and like if you talk with parents, a lot of them will tell you that, oh yeah, if they give their kids they give their kids who have an autism diagnosis melatonin to help them go to sleep, and otherwise it's very, very hard to get for them to go to sleep. Yeah, and so there's a number of these things these co-occurring conditions that are much overrepresented in autism. Yeah, so and please feel free to interrupt me if I'm getting too much off topic here. But we spend a significant amount of time on saying, okay, rather than people just saying people just have an autism diagnosis, can I actually find, are there some subgroups of people with the autism diagnosis, particularly subgroups that are defined by the co-occurring conditions, both which co-occurring conditions they have, as well as when did they appear during development. Yeah, because I'm not completely convinced that everybody, well, actually, I'm far from convinced that everybody with the autism diagnosis has the same thing. They all have a diagnosis, but biologically, there's probably several different things that all are underlying this. Yeah. And but when I start basically finding subgroups, then hopefully these groups aren't as heterogeneous as you find with just the autism diagnosis. Okay. And so what we did is we basically, for example, we looked in one study at children from birth to age 5 and knew that, well, at some point during that time, if they got an autism diagnosis, what other conditions were they diagnosed with? Yeah, like say, for example, were they checked out for GI problems? Were they checked for epilepsy? Yeah, and it was, so we basically, so we did a study on this, and what we found, using a clustering algorithm, we came up with there's 3 significant subgroups, yeah, in this space, based upon co-occurring conditions. And I think that that's, for example, one way to look at this, because the co-occurring conditions you can, or at least many of them, you can treat and they improve people's lives tremendously. And so this is actually something that I would argue can certainly help people. Yeah. Another thing we did related to this study, we looked at risk factors during pregnancy and said what risk factors will result in an autism diagnosis later. And one interesting thing is we took this study where I found the 3 subgroups based upon co-occurring conditions and saying, what are the risk factors? It turns out the risk factors for each one of those 3 groups is totally different. Yeah, which is another indicator that biologically you have many different things that can underlie this that all ultimately may end up in an autism diagnosis. But clearly somebody who has autism and GI problems, you can try to do something about the GI problems, but this would be different from somebody who has autism and epilepsy, how you look at this. Yeah, and so this idea to look at the co-occurring conditions that happen at some point during the lifetime is really important. Our work mainly focuses on early life, yeah, but there are clearly other things where you may find that co-occurring conditions develop later in life, and there's a lot of interesting work to be done there. That's just not what we've done so far.
Is there any exciting project or research you're currently working on that you'd like to share?
So, well, I said a few of the key things is there's using basically data science on blood metabolite measurements for autism diagnosis is certainly one thing we're working on, we're interested in. Likewise, we also view this that instead of just saying does somebody have an autism diagnosis or not, but can we use the same thing to determine like what's the severity with regard to certain measurements? Let's say, for example, adaptive behavior is one item that often comes up. Can I can I predict the severity of the adaptive behavior score based upon blood measurements? This then opens up the opportunity if somebody wants to modify either lifestyle or undergo some treatment, will the changes I'm seeing in the blood metabolites also predict the changes you're seeing in adaptive behavior, for example? So you view this as a companion diagnostic, because the current state of the art is always that you ask people, well, how are you feeling now? And after the treatment, how are you feeling afterwards? But oftentimes there's like 6 months in between and you can't remember, did I score a 7 or 8 out of a 10 point scale 6 months ago? And then you're looking for minor differences. So looking at blood metabolites, you have an objective measurement that doesn't change. So that's another thing. Developing this further into companion diagnostic is something we're interested in. I did mention the work on the co-occurring conditions and the risk factors. I think getting some more structure into what does the autism diagnosis mean biologically is a really important topic. That's something we're working on. Another topic that's the big item for us, but we're just the ones running the analysis on this is you're looking at the microbiota. Yeah, this is mainly focused on what happens if Now, what happens? Can I change the microbiome to basically improve GI symptoms that people have? We are involved in several studies on this, but as I said, we run some analysis. I don't run these studies, so my group is secondary there. So it's not really, we're not the leaders on that, but it's something we're definitely involved in. Last item, we're working with a residential facility, and they're interested in, can I take data over time from every individual, and does it allow me to make predictions? For example, is like, what's your likelihood of somebody exhibiting a challenging behavior or not, based upon this data over time, because it allows them to better plan things. And it's in, I mean, there's no biology in that part of it, but it's really, we're looking at behavioral data over time, as well as other medical data you collect from somebody, and can I make a prediction of the likelihood of a challenging behavior occurring or not? So this is that's work in progress. Okay, you would have to watch our website and read our papers in the future to see where this is going, but it's certainly an interesting project. In a nutshell, that kind of summarizes what we're working on right now.
How do you see the role of artificial intelligence and machine learning evolving in analyzing biological data? Do you foresee any specific challenges or opportunities in integrating these technologies with biomedical research?
Oh, look, they play a huge role. Okay, it's most apparent any type of biomedical imaging. Because ultimately the artificial intelligence looks at identifying certain things in the image and tells you what to do from there. So it plays a big role. That, by the way, my group does not work on the imaging side, but given that I'm the department head and I have several here and I have several faculty who work in this field, that's definitely a very big item already. But more generally, finding patterns as that we focus on metabolites, that doesn't mean that there's other omics data people can look at. That's also interesting. You're going to need AI and machine learning type of methods to sort through this to find patterns. So big role there. With regards to challenges, I kind of hinted at this earlier. Like a lot of the AI and machine learning algorithms, they were developed for, or at least, let's say, they were made more popular. For the use in for like search engines and self-driving cars and these types of things. That's not what we're using them for. Okay, I mean, obviously, if somebody looks at an image and there's some scan of parts of the body on this image, that's very different than identifying an object for a self-driving car. The algorithm, there may be similarities, but you're going to have to adjust this. Let's say the biggest item that I always see in this, okay, is we are limited really by the number of patients that we have. And let me give you an example. Like traditionally, when you use these techniques, if I have data from, or if I'm looking at, let's say, measuring one metabolite, I would like to have data from 10 to 20 patients in order to identify a coefficient that tells me how important this metabolite is. Okay, so meaning if I use 5 metabolites, then I would like data from 50 to 100 patients. The problem we're running into nowadays is that we can take many, many measurements from the same blood sample from the same patient. So I have samples where we got 4,000 or 5,000 measurements back from one single sample. Now, if I were to look at all these 4,000 or 5,000 and I want 10 to 20 patients for this, that means I would have to, look at tens of thousands of patients in a study. You cannot run a medical study like this. Most medical studies, if they're phase 1, they're 20, 30 people. If they're phase 2, maybe they're 40 to 80 or something like this. And if they're phase 3, they're a few hundred. Maybe a few thousand at most in the most extreme cases. But so this ratio of how many patients I have per measurement is just simply not there. The ratio is the opposite of what you would like it to be. And so you have to adapt your algorithms to make sure that how can I still live in this realm when the methods that I'm using are developed for having basically very different patient to measurement ratio. And so that's a really big thing to look at. And that's when people ask, so what is your group doing that Yeah, not like anybody else could do this, right? If I just give them the algorithm. And these are kind of questions that we are dealing with. How do I effectively deal with this? Because the one thing that you always want to make sure is that I don't just want to find out that my prediction worked on the one data set that you gave me. I'm asking, well, if you give me different data sets but trying to look for the same thing, will my predictions hold up? And that's what you want to make sure that your predictions are robust. And you really have to focus on, when you use these algorithms, on how to go about this, because you can get very misleading results where you get almost perfect prediction on the one data set, and then it doesn't generalize to the next one that comes in. And that's very detrimental. I mean, it gets people very disappointed and starting to wonder, about the meaning of all the work you've did. So it's very important to get that right from the start.
Your work, which involves creating these algorithms to analyze large-scale biological data. So regarding that, how do you ensure the accuracy and reliability of these models, especially when dealing with high throughput data from genomic proteomics?
Yeah, and so this is really this is a great question. There's several ways to go about this, but part of it is you make predictions on what you have your training set that your model and your classifiers were trained on, but you also always have to leave some samples out and make sure that you can predict those, even though your model has not seen them before. So that's a really important thing. What is then somewhat special about our work is that we never have enough data. Okay, I have way too many or almost too many measurements per individual, but I never have enough individuals. No matter how many you give me, it will never be enough. Okay, but so I have to live with whatever I can get. One of the things you do is that, for example, you use a Monte Carlo approach. We're saying, let's repeat this. Let's take our data set, split some of part of the data set off, train the model on the parts that you used, predict the part that was split off that you haven't seen before, and then see how accurate you are. Repeat the entire process. Okay, randomly split a different part up, identify a new model, tell me what prediction you have, and then I statistically analyze after you've done this thousands of times. So how accurate were you across all of this? How much did your model structure change as you randomly use different training test splits and things like this? You need to do something along those lines, or as a minimum, something called leave 1, 2, or how many ever out cross-validation, because otherwise you don't have the robustness of your prediction. And to put it bluntly, anybody can get you good prediction accuracy. There's no value in this. I can get you 100% prediction accuracy for any sample you give me. But you want the fitting accuracy. What you want is the prediction on a validation set accuracy. That's the key. That's the one that shows that there's robustness in there, and that's usually much, much lower. And that you have to prevent what's called overfitting the model. Okay, and so that's what we spent a lot of time on. And the methods I just described, they help you with making sure you are not overfitting your models.
As the department head of biomedical engineering, how do you encourage students to think interdisciplinarily and apply engineering principles to solve real-world biological challenges?
So several things. I mean, one thing is, and this is, by the way, a thing that RPI in general is famous for, we often do work at the interface of disciplines. So this whole idea of interdisciplinary work is kind of woven into our DNA, and the students we attract are usually interested in this from the start. So it helps if people have a certain predisposition for that coming in. But the other thing is, I tell them, look, for the work that we do, if I go to just the systems community and present to them what we do, they tell me, well, on the algorithm side, there's nothing new here. And they're right. If I talk to people who are just doing autism research, they're telling me, well, you do all these funny things with algorithms. I'm not sure there's the value, and it's very different from how it's traditionally diagnosed, and so I'm not sure there is a lot of value. But by working right at the interface, you are able to suddenly come up with solutions that, in just staying in either of the two realms, you're not going to do. Yeah, it's basically because this application of these techniques, even though they may seem standard to the systems folks, but having them applied to a neurodevelopmental condition like autism is something that is new. And there's a lot of value in this. And this is what drives discovery forward. So that's certainly, and I use this as an example to tell people, look, oftentimes that's where you make the most interesting discoveries. So you have to work interdisciplinary. The last thing is, and I said, that's related to my new job. So I'm the director of the Center for Biotechnology and Interdisciplinary Studies. That's the name of the center. Okay, so it's even reflected in the name of our center that we are focusing on bringing people from different fields together to jointly work on problems to really make some headway at the interface of different disciplines. And this is just one example.
What kind of advice would you give to young researchers or students looking to enter the field of computational biology and biomedical engineering? What skills or knowledge are most important for success and, conducting good research in the area?
I mean, look, the first thing is, if you want to do this type of work, you have to like computational work. You have to like sitting in front of a computer, because that's what you're going to do most of the day. You have to like some form of programming. I don't care which language, but formulating algorithms, yeah, and seeing how they work is something that you have to have some passion for. Yeah, we program most of our algorithms ourselves. Okay, I mean, yeah, we may use some subroutines that exist, in particular in Python and data science realm, but we are writing code really to analyze these data. This isn't just let's click a button and some program analyzes this for us. You have to tailor this. So you have to like sitting in front of a computer writing algorithms. You also have to like trying out different things. I mean, that's the nice thing about computational work. Yeah, you can try and make some modifications, see what to get. It didn't cost you anything. It cost you a little bit of time. Yeah, but in experimental work, you may have suddenly, by made some change, ruined an experiment that cost you thousands or tens of thousands of dollars. For us, it doesn't. It's some computational time if it doesn't work. Yeah, and so you have to try different things, because trust me, when you get the result the first time, nobody believes it. Okay, you have to always validate it, and you think about other computational experiments, how to validate it. So you have to like to do this type of problem solving in addition to that. So those are the kind of things, and ultimately, I mean, I personally find it a very rewarding area to work in, because you have applications that directly actually affect people's lives, or at least they have the potential to do that. And so you have to have some passion for the application too. Okay, so somebody works on rocket science that can be very excited about it, but that may not necessarily be the right person who wants to work in biomedical engineering, where you're dealing with people. You have to, you have to have some passion for the application in addition to just the algorithms. Yeah, but I think you need a bit of both. Okay, like if somebody just wants to program, that's nice, but they may be better off in computer science because ultimately you're dealing with data that comes from patients, and these patients usually have health conditions. That's why you have the data, and you have to have some interest in saying, yeah, this is a problem I want to solve, or at least to contribute to the solution of it.
Could you tell us about your future career plans and what we can expect from your new role in 2025?
As I mean, first of all, career plans. I literally just a month ago started this new position. As a center director. I'm still at RPI. My research group is not going to change. In fact, my research group was already in the center that I'm now heading, so that doesn't change much. With regard to research, honestly, the problems I kind of outlined, they'll keep us busy for a long time. Yeah, there's many subtopics within those realms that we definitely want to explore, and I think this will keep us busy for several years. So I'll leave it at that.
Then, Dr. Hahn, thank you for taking the time to share your insights and expertise. Your groundbreaking work in systems biology and autism research is really inspiring, and it's really amazing to see how your innovative approaches are actually advancing science and healthcare. So, that's I guess, a really strong, powerful example of how engineering and medicine can come together to tackle complex challenges. So thank you once again for your time and for sharing your perspective.
Thank you for having me.